Chapter 3: Energy, Catalysis, and Biosynthesis. One property above all makes living things seem almost miraculously different from non-living matter. They create and maintain order in a universe that is tending always toward greater disorder. To create this order, the cells in a living organism must carry out a never-ending stream of chemical reactions. In some of these reactions, small organic molecules, amino acids, sugars, nucleotides, and lipids are taken apart or modified to supply the many other small molecules that the cell requires. In other reactions, these small molecules are used to construct an enormously diverse range of proteins, nucleic acids, and other macromolecules that endow living systems with all of their most distinctive properties. Each cell can be viewed as a tiny chemical factory, performing many millions of reactions every second. To carry out the many chemical reactions needed to sustain it, a living organism requires both a source of atoms in the form of food molecules and a source of energy. The atoms and the energy must both come ultimately from the non-living environment. In this chapter, we discuss why cells require energy and how they use energy and atoms from their environment to create the molecular order that makes life possible. Many of the chemical reactions that cells perform would normally occur only at temperatures that are much higher than those inside a cell. Each reaction, therefore, requires a major boost in chemical reactivity to enable it to proceed rapidly within the cell. This boost is provided by specialized proteins called enzymes, each of which accelerates or catalyzes just one of the many possible kinds of reactions that a particular molecule might undergo. These enzyme-catalyzed reactions are usually connected in series, so that the product of one reaction becomes the starting material for the next. Figure 3.1. The long linear reaction pathways, or metabolic pathways that result, are in turn linked to one another, forming a complex web of interconnected reactions. Rather than being an inconvenience, the necessity for catalysis is a benefit, as it allows the cell to precisely control its metabolism, the sum total of all the chemical reactions it needs to carry out to survive, grow, and reproduce. This control is central to the chemistry of life. Two opposing streams of chemical reactions occur in cells: the catabolic pathways and the anabolic pathways. The catabolic pathways, catabolism, break down foodstuffs into smaller molecules, thereby generating both a useful form of energy for the cell and some of the small molecules that the cell needs as building blocks. The anabolic or biosynthetic pathways, anabolism, use the energy harnessed by catabolism to drive the synthesis of the many molecules that form the cell. Together, these two sets of reactions constitute the metabolism of the cell. Figure 3.2. The details regarding the individual reactions that comprise cell metabolism are part of the subject matter of biochemistry, and they need not concern us here, but the general principles by which cells obtain energy from their environment and use it to create order are central to cell biology. We begin this chapter with a discussion of why a constant input of energy is needed to sustain living organisms. We then discuss how enzymes catalyze the reactions that produce biological order. Finally, we describe the molecules that carry the energy that makes life possible. The use of energy by cells. Non-living things left to themselves eventually become disordered. Buildings crumble and dead organisms decay. Living cells, by contrast, not only maintain but actually generate order at every level, from the large-scale structure of a butterfly or a flower down to the organization of the molecules that make up these organisms. Figure 3.3. This property of life is made possible by elaborate cellular mechanisms that extract energy from the environment and convert it into the energy stored in chemical bonds. Biological structures are therefore able to maintain the form even though the materials of which they are made are continually being replaced and recycled. Your body has the same basic structure it had 10 years ago, even though you now contain atoms that, for the most part, were not in your body then. Biological order is made possible by the release of heat energy from cells. The universal tendency of things to become disordered is expressed in a fundamental law of physics, the second law of thermodynamics. This law states that in the universe or in any isolated system, a collection of matter that is completely isolated from the rest of the universe, the degree of disorder can only increase. The second law of thermodynamics has such profound implications for living things that it is worth restating it in several ways. We can express the second law in terms of probability by stating that systems will change spontaneously toward those arrangements that have the greatest probability. Consider a box of 100 coins all lying heads up. A series of events that disturbs the box will tend to move the arrangement towards a mixture of 50 heads and 50 tails. The reason is simple. There are a huge number of possible arrangements of the individual coins that can achieve the 50-50 result, but only one possible arrangement that keeps them all oriented heads up. Because the 50-50 mixture accommodates a greater number of possibilities and places fewer constraints on the orientation of each individual coin, we say that it is more disordered. For the same reason, one's living space will become increasingly disordered without an intentional effort to keep it organized. Movement toward disorder is a spontaneous process, requiring a periodic effort to reverse it. Figure 3.4. The measure of a system's disorder is called the entropy of the system, and the greater the disorder, the greater the entropy. Thus, another way to express the second law of thermodynamics is to say that systems will change spontaneously towards arrangements with greater entropy. Living cells, by surviving, growing, and forming complex communities and even whole organisms, are generating order, and thus might appear to defy the second law of thermodynamics. This is not the case, however, because a cell is not an isolated system. Rather, it takes in energy from its environment in the form of food, inorganic molecules, or photons of light from the sun, and it then uses this energy to generate order within itself, forging new chemical bonds or building large macromolecules. In the course of performing the chemical reactions that generate order, chemical bond energy is converted into heat. Heat is energy in its most disordered form, the random jostling of molecules, analogous to the random jostling of the coins in the box. Because a cell is not an isolated system, the heat energy that its reactions generate is quickly dispersed into the cell's surroundings. There, the heat increases the intensity of the thermal motions of the resident molecules, thereby increasing the entropy of the environment. Figure 3.5. The amount of heat released by a cell must be great enough that the order generated inside the cell is more than compensated for by the decrease in order in the environment. Only in this case is the second law of thermodynamics satisfied, because the total entropy of the system, that of the cell plus its environment, will increase as a result of the chemical reactions inside the cell. Where does the heat released by the cell come from? According to the first law of thermodynamics, energy can be converted from one form to another, but it cannot be created or destroyed. The interconversions of some forms of energy are illustrated in Figure 3.6. The amount of energy present in different forms will change as a result of the chemical reactions inside the cell, but the first law tells us that the total amount of energy in the universe must always be the same. For example, as an animal cell breaks down foodstuffs, some of the energy in the chemical bonds in the food molecules, chemical bond energy, is converted into the thermal motion of molecules, Heat energy. This conversion of chemical energy into heat energy is essential if the reactions inside the cell are to cause the universe as a whole to become more disordered, as required by the second law. The cell cannot derive any benefit from the heat energy it produces. However, unless the heat-generating reactions inside the cell are directly linked to processes that maintain molecular order. It is the tight coupling of heat production to an increase in order that distinguishes the metabolism of a cell from the wasteful burning of fuel in a fire. Later in this chapter, we shall illustrate how this coupling occurs. For the moment, it is sufficient to recognize that, by direct linking the burning of food molecules to the generation of biological order, cells are able to create and maintain an island of order in a universe tending toward chaos. Photosynthetic organisms use sunlight to synthesize organic molecules. All animals live on energy stored in the chemical bonds of organic molecules made by other organisms, which they take in as food. These food molecules also provide the atoms that animals need to construct new living matter. Some animals obtain their food by eating other animals. At the bottom of the animal food chain, however, are animals that consume plants or other photosynthetic organisms. Before being eaten, these organisms trap energy from sunlight, so the energy animals obtained by eating plants came originally from the sun. Figure 3.7. Solar energy enters the living world through photosynthesis, a process that converts the electromagnetic energy in sunlight into chemical bond energy in cells. Photosynthetic organisms, including plants, algae, and some bacteria, are able to obtain all of the atoms they need from inorganic sources. Plants, for example, use carbon from atmospheric carbon dioxide, hydrogen and oxygen from water, nitrogen from ammonia, and nitrates in the soil, and other elements needed in smaller amounts from inorganic salts in the soil. They use the energy they derive from sunlight to form chemical bonds between these atoms, linking them into small chemical building blocks such as sugars, amino acids, nucleotides, and fatty acids. These small molecules in turn are converted into the macromolecules, the proteins, nucleic acids, polysaccharides, and lipids that form the plant. All of these substances serve as nutrients for animals, fungi, and non-photosynthetic bacteria that may later feed on the plant. The reactions of photosynthesis take place in two stages, one that depends on light and another that does not. Figure 3.8. In the first light-dependent stage, energy from sunlight is captured and transiently stored as chemical bond energy in specialized small molecules that carry energy in their reactive chemical groups. We discuss these activated carrier molecules in more detail later in the chapter. Molecular oxygen, O2 gas, derived from the splitting of water by light, is released as a byproduct of this first stage. In the second stage of photosynthesis, the molecules that serve as energy carriers are used to help drive a carbon fixation process in which sugars are manufactured from carbon dioxide gas, CO2, and water, H2O. By producing sugars, these light-independent reactions generate an essential source of stored chemical bond energy and materials, both for the plant itself and for any animals that eat it. We describe the elegant mechanisms that underlie these two stages of photosynthesis in detail in Chapter 14. The net result of both stages of photosynthesis, as far as a green plant is concerned, can be summarized simply in the equation light energy plus CO2 plus H2O equals sugars plus O2 plus heat energy. The sugars produced are then used both as a source of chemical bond energy and as a source of materials to make the many other small and large organic molecules that are essential to the plant cell. Cells obtain energy by the oxidation of organic molecules. All animals and plant cells require the chemical energy stored in the chemical bonds of organic molecules, either the sugars that a plant has photosynthesized as food for itself, or the mixture of large and small molecules that an animal has eaten. To use this energy to live, grow, and reproduce, organisms must extract it in a usable form. In both plants and animals, energy is extracted from food molecules by a process of gradual oxidation, or controlled burning. Earth's atmosphere is about 21% oxygen, and in the presence of oxygen, the most energetically stable form of carbon is CO2, and that of hydrogen is H2O. A cell is therefore able to obtain energy from sugars or other organic molecules by allowing the carbon and hydrogen atoms in these molecules to combine with oxygen, that is, become oxidized, to produce CO2 and H2O respectively, a process known as cellular respiration. Photosynthesis and respiration are complementary processes. Figure 3.9. This means that the transaction between plants and animals are not all one way. Plants, animals, and microorganisms have existed together on this planet for so long that many of them have become dependent and essential part of each other's environments. The oxygen released by photosynthesis is consumed by nearly all organisms for the combustion of organic molecules. And some of the CO2 molecules that are fixed today into organic molecules by photosynthesis in a green leaf were released yesterday into the atmosphere by the respiration of an animal, fungus, or bacterium decomposing dead organic matter, or by the respiration of the plant itself. Carbon utilization therefore forms a huge cycle that involves the biosphere, all of the living organisms on Earth as a whole, crossing boundaries between individual organisms. Figure 3.10. Similarly, atoms of nitrogen, phosphorus, and sulfur move between the living and non-living worlds in cycles that involve plants, animals, fungi, and bacteria. Prokaryotes, in fact, are estimated to contain nearly half the carbon stored in living organisms, and they are the single largest reservoir of nitrogen and phosphorus on Earth, containing 10 times more of these nutrients than plants. Oxidation and reduction involve electron transfers. The cell does not oxidize organic molecules in one step, as occurs when organic material is burned in a fire. Through the use of enzyme catalysts, metabolism carries the molecules through a large number of reactions that only rarely involve the direct addition of oxygen. Before we consider some of these reactions and the purpose behind them, we need to explain what is meant by oxidation. The term oxidation literally means the addition of oxygen atoms to a molecule. More generally, though, oxidation is said to occur in any reaction in which electrons are transferred from one atom to another. Oxidation, in this sense, refers to the removal of electrons. The converse reaction, called reduction, involves the addition of electrons. Thus, Fe2+ is oxidized when it loses an electron to become Fe3+, whereas a chlorine atom is reduced when it gains an electron to become Cl-. Because the number of electrons is conserved in a chemical reaction, there is no net loss or gain. Oxidation and reduction always occur simultaneously. That is, if one molecule gains an electron in a reaction, reduction, a second molecule must lose the electron, oxidation. When a sugar molecule is oxidized to CO2 and H2O, for example, the O2 molecules involved in forming H2O gain electrons and thus are said to have been reduced. The terms oxidation and reduction apply even when there is only a partial shift of electrons between atoms linked by a covalent bond. When a carbon atom becomes covalently bonded to an atom with a strong affinity for electrons, oxygen, chlorine, or sulfur for example, it gives up more than its equal share of electrons and forms a polar covalent bond. The positive charge of the carbon nucleus now slightly exceeds the negative charge of its electrons. Thus, the carbon atom acquires a partial positive charge and is said to be oxidized. Conversely, the carbon atom in a CH bond has somewhat more than its share of electrons. It acquires a partial negative charge, and so is said to be reduced. Figure 311a. When a molecule in a cell picks up an electron, it often picks up a proton at the same time, protons being freely available in water. The net effect in this case is to add a hydrogen atom to the molecule. A plus electron plus proton equals AH. Even though a proton plus an electron is involved, instead of just an electron, such hydrogenation reactions are reductions, and the reverse, dehydrogenation reactions, are oxidations. An easy way to tell whether an organic molecule is being oxidized or reduced is to count its CH bonds. Reduction occurs when the number of CH bonds increases, whereas oxidation occurs when the number of CH bonds decreases. Figure 311b. As we shall see later in this chapter, cells use enzymes to catalyze the oxidation of organic molecules in small steps, through a sequence of reactions that allows useful energy to be harvested. Free energy and catalysis. Enzymes, like cells, obey the second law of thermodynamics. Although they can speed up energetically favorable reactions, those that produce disorder in the universe, enzymes cannot by themselves force energetically unfavorable reactions to occur. Cells, however, must do just that in order to grow and divide. They must build highly ordered and energy-rich molecules from small and simple ones, a process that requires an input of energy. To understand how enzymes can catalyze such energetically unfavorable but life-sustaining reactions, we first need to examine the energetics involved. In this section, we consider how the free energy of molecules contributes to their chemistry, and we see how free energy changes, which reflect how much disorder is generated by a reaction, influence whether and how that reaction will proceed. We also discuss how enzymes exploit differences in free energy changes to produce biological order. But first, we shall review how catalysis occurs rapidly and with precision inside living cells. Enzymes lower the energy barrier that prevent chemical reactions from occurring. Paper burns readily, releasing into the atmosphere water and carbon dioxide as gases and energy as heat. Paper plus O2 equals smoke plus ash plus heat plus CO2 plus H2O. But this occurs in only one direction. Smoke and ashes never spontaneously gather carbon dioxide and water from the heated atmosphere and reconstitute themselves into paper. When paper burns, its chemical energy is dissipated as heat, not lost from the universe, since energy can never be created or destroyed, but irretrievably dispersed in the chaotically random thermal motions of molecules. At the same time, the atoms and molecules of the paper become dispersed and disordered. In the language of thermodynamics, there has been a release of free energy, that is, of energy that can be harnessed to do work or drive chemical reactions. This release reflects a loss of orderliness in the way the energy and molecules had been stored in the paper. We will discuss free energy in more detail shortly, but the general principle can be summarized as follows. Chemical reactions proceed only in the direction that leads to a loss of free energy. In other words, the spontaneous direction for any reaction is the direction that goes downhill. A downhill reaction, in this sense, is said to be energetically favorable. Although the most energetically favorable form of carbon under ordinary conditions is CO2, and that of hydrogen is H2O. A living organism will not disappear in a puff of smoke, and the book in your hands will not burst spontaneously into flames. This is because the molecules in both the living organism and the book are in a relatively stable state, and they cannot be changed to lower energy states without an initial input of energy. In other words, a molecule requires a boost over an energy barrier before it can undergo a chemical reaction that moves it to a lower energy, more stable state. Figure 312a. This boost is known as the activation energy. In the case of a burning book, the activation energy is provided by the heat of a lighted match. For the molecules in the watery solution inside a cell, the boost is delivered by an unusually energetic random collision with surrounding molecules, collisions that become more violent as the temperature is raised. At the temperatures in living cells, the push over the energy barrier is greatly aided by specialized proteins called enzymes. Each enzyme binds tightly to one or two molecules, called substrates, and holds them in a way that greatly reduces the activation energy needed to facilitate a specific chemical reaction between them. Figure 312b. A substance that can lower the activation energy of a reaction is termed a catalyst. Catalysts increase the rate of chemical reactions because they allow a much larger proportion of the random collisions with surrounding molecules to kick the substrates over the energy barrier, as illustrated in Figure 313 and Figure 314a. Enzymes are among the most effective catalysts known. They can speed up reactions by a factor of as much as 10 to the power 14, that is, trillions of times faster than the same reactions would proceed without an enzyme catalyst. Enzymes, therefore, allow reactions that would not otherwise occur to proceed rapidly at normal temperatures. Without enzymes, life could not exist. Unlike temperature, enzymes are highly selective. Each enzyme usually speeds up only one particular reaction out of the several possible reactions that its substrate molecules could undergo. In this way, enzymes direct each of the many different molecules in a cell along specific reaction pathways, Figure 314b and c, thereby producing the compounds that the cell actually needs. The success of living organisms can be attributed to the cell's ability to make enzymes of many types, each with precisely specified properties. Each enzyme has a unique shape containing an active site, a pocket or groove in the enzyme into which only particular substrates will fit, Figure 315. Like all catalysts, enzyme molecules themselves remain unchanged after participating in a reaction, and therefore can function over and over again. In Chapter 4, we will discuss further how enzymes work, after we have looked in detail at the molecular structure of proteins. The free energy change for a reaction determines whether it can occur. According to the second law of thermodynamics, a chemical reaction can proceed only if it results in a net or overall increase in the disorder of the universe. See Figure 3.5. Disorder increases when useful energy that could be harnessed to do work is dissipated as heat. The criterion for an increase of disorder can be expressed most conveniently in terms of the free energy, G, of a system. The value of G is of most interest when a system undergoes a change, so the free energy change, denoted delta G, is the term we most often see. Suppose that the system being considered is a collection of molecules. Because of the way free energy is defined, delta G measures the amount of disorder created in the universe when a reaction takes place that involves these molecules. Energetically favorable reactions, by definition, are those that create disorder by decreasing the free energy of the system to which they belong. In other words, they have a negative delta G. Figure 316. A reaction can occur spontaneously only if delta G is negative. On a macroscopic scale, a familiar example of an energetically favorable reaction with a negative delta G is the reaction by which a compressed spring relaxes to an expanded state, releasing its stored elastic energy as heat to its surroundings. An example on a microscopic scale is the dissolving of salt in water. Conversely, energetically unfavorable reactions with a positive delta G, such as those in which two amino acids are joined together to form a peptide bond, by themselves create order in the universe. These reactions cannot occur spontaneously. Energetically unfavorable reactions can take place only if they are coupled to a second reaction with a negative delta G so large that the net delta G of the entire process is negative. Figure 317. Life is possible because enzymes can create biological order by coupling energetically unfavorable reactions with energetically favorable ones. These critical concepts are summarized with examples in panel 3.1, pages 94 to 95. The concentration of reactants influences the free energy change and a reaction's direction. As we have just described, a reaction Y to X will go in the direction Y to X when the associated free energy change, delta G, is negative, just as a tensed spring left to itself will relax and lose its stored energy to its surroundings as heat. For a chemical reaction, however, delta G depends not only on the energy stored in each individual molecule, but also on the concentrations of the molecules in the reaction mixture. Remember that delta G reflects the degree to which a reaction creates a more disordered, in other words, a more probable state of the universe. Recalling our coin analogy, more coins in a jiggling box will flip from a head to a tail orientation when the box contains 90 heads and 10 tails, than when the box contains 10 heads and 90 tails. The same is true for a chemical reaction. For the reversible reaction Y to X, X to Y, a large excess of Y over X will tend to drive the reaction in the direction Y to X. That is, there will be a tendency for there to be more molecules making the transition Y to X than there are molecules making the transition X to Y. Thus, as the ratio of Y to X increases, the delta G becomes more negative for the transition Y to X and more positive for the transition X to Y. The standard free energy change makes it possible to compare the energetics of different reactions. Because delta G depends on the concentrations of the molecules in the reaction mixture at any given time, it is not a particularly useful value for comparing the relative energies of different types of reactions. Such comparisons are necessary, for example, to predict whether an energetically favorable reaction is likely to have a delta G negative enough to drive an energetically unfavorable reaction. To level the playing field and place reactions on a comparable basis, we need to turn to the standard free energy change of a reaction, delta G degrees. The delta G degrees is independent of concentration. It depends only on the intrinsic characters of the reacting molecules, based on their behavior under ideal conditions, where the concentrations of all the reactants are set to the same fixed value of 1 mole per liter. For the reaction Y to X at 37 degrees Celsius, delta G degrees is related to delta G as follows. Delta G equals delta G degrees plus RT ln of X over Y, where delta G is in kilocalories per mole. Concentration of Y and concentration of X denote the concentrations of Y and X in moles per liter. Ln is the natural logarithm, and RT is the product of the gas constant R and the absolute temperature T. At 37 degrees Celsius, RT equals 0.616. A mole is 6 times 10 to the power of 23 molecules of a substance. A large body of thermodynamic data has been collected from which delta G degrees can be calculated for most metabolic reactions. Some common reactions are compared in terms of their delta G degrees in panel 3.1, pages 94 to 95. Cells exist in a state of chemical disequilibrium. From the equation above, we can see that when the molar concentrations of Y and X are equal, that is, concentration of X over concentration of Y equals 1, the value of delta G equals the value of delta G degrees, because ln of 1 equals 0. But as the favorable reaction Y to X proceeds, the concentration of the product X increases and the concentration of the substrate Y decreases. This change in relative concentrations will cause the concentration of X over concentration of Y to become larger, making the initially favorable delta G less and less negative. The natural logarithm is positive for a number more than 1 and negative for a number less than 1. Chemical reactions will generally proceed until they reach a state of equilibrium. At that point, the rates of the forward and reverse reactions are equal, and there is no further net change in the concentrations of substrate or product. Figure 318. For reactions at chemical equilibrium, delta G equals 0, so the reaction will not proceed forward or backward, and no work can be done. Because the maintenance of order within a cell requires a continuous input of energy, any cell whose reactions have all reached chemical equilibrium is dead. Living cells avoid reaching a state of equilibrium because they are constantly exchanging materials with their environment, taking in nutrients and eliminating waste products. Many of the individual reactions in a cell's complex metabolic network are kept in disequilibrium because the products of one reaction are continually being siphoned off to become the substrates in a subsequent reaction. Rarely do products and substrates reach concentrations at which the forward and reverse reaction rates are equal. The equilibrium constant is directly proportional to delta G degrees. As we have seen, at chemical equilibrium, when the forward and reverse reaction rates are equal, the ratio of substrate to product will remain constant. This state makes it possible to calculate a reaction's equilibrium constant, K. K equals concentration of X over concentration of Y, where concentration of X is the concentration of the product and concentration of Y is the concentration of the reactant at equilibrium. This expression describes the situation at the point at which the concentration effect just balances the push given to the reaction by delta G degrees, so that delta G equals 0, and there is no net change of free energy to drive the reaction in either direction. See panel 3.1, page 95. So because delta G equals delta G degrees plus RT ln of concentration of X over concentration of Y, as stated on page 92, then at equilibrium at 37 degrees Celsius, where delta G equals 0 and RT equals 0.616, the equation becomes delta G degrees equals negative 0.616 times ln of concentration of X over concentration of Y, or delta G degrees equals negative 0.616 times ln of K. Converting this equation from natural log ln to the more commonly used base 10 logarithm log, we get delta G degrees equals negative 1.42 log K. This equation reveals how the equilibrium ratio of Y to X, expressed as the equilibrium constant K, depends on the intrinsic character of the molecules, as expressed in the value of delta G degrees. Table 3.1. Note that for every 1.42 kcal per mole difference in free energy at 37 degrees Celsius, the equilibrium constant changes by a factor of 10. Thus, the more energetically favorable the reaction, the more product will accumulate if the reaction proceeds to equilibrium. In complex reactions, the equilibrium constant depends on the concentrations of all reactions and products. We have so far discussed the simplest of reactions, Y to X, in which a single substrate is converted into a single product. But what happens in the more common situation where two reactants combine to form a single product, A plus B to AB and AB to A plus B? The same principles apply, except that now the equilibrium constant K depends on the concentrations of both of the reactants and that of the product. K equals concentration AB over concentration of A times concentration of B. The concentrations of both substrates are multiplied because the formation of product AB depends on the collision of A and B, and these encounters occur at a rate that is proportional to concentration of A times concentration of B. As with single substrate reactions, delta G degrees equals negative 1.42 log K at 37 degrees Celsius. The equilibrium constant indicates the strength of molecular interactions. The concept of free energy change does not apply to chemical reactions where covalent bonds are being broken and formed, but also to interactions where one molecule binds to another by means of non-covalent bonds. See Chapter 2, pages 62 to 63. These types of interactions are immensely important to cells. They include the binding of substrates to enzymes, the binding of gene regulatory proteins to DNA, and the binding of one protein to another to make the myriads of different structural and functional protein complexes that constitute a living cell. Two molecules will bind to each other if the delta G degrees of the interaction is negative. That is, the free energy of the resulting complex is lower than the sum of the free energies of the two partners when unbound. Because the equilibrium constant of a reaction is related directly to delta G degrees, K is commonly employed as a measure of the binding strength of a non-covalent interaction between two molecules. The binding strength is a very useful quantity to know because it also indicates how specific the interaction is between the two molecules. Consider the reaction shown in Figure 319, where molecule A interacts with molecule B to form the complex AB. The reaction proceeds until it reaches equilibrium, at which point the number of association events precisely equals the number of dissociation events. At this point, the concentration of reactants and of the complex AB can be used to determine the equilibrium constant. K becomes larger as the binding energy, that is, the energy released in the binding interaction, increases. In other words, the larger K is, the greater is the drop in free energy between the dissociated and associated states, and the more tightly the two molecules will bind. Even a change of a few non-covalent bonds can have a striking effect on a binding interaction, as illustrated in Figure 320. In this example, eliminating a few hydrogen bonds from a binding interaction can be seen to cause a dramatic decrease in the amount of complex that exists at equilibrium. For sequential reactions, the changes in free energy are additive. Now we return to our original concern. How can enzymes catalyze reactions that are energetically unfavorable? One way they do so is by directly coupling energetically unfavorable reactions with energetically favorable ones. Consider, for example, two reactions, X to Y and Y to Z, where the delta G degree values are plus 5 and negative 13 kcal per mole respectively. Recall that a mole is 6 times 10 to the power of 23 molecules of a substance. The unfavorable reaction X to Y will not occur spontaneously. However, it can be driven by the favorable reaction Y to Z, provided that the second reaction follows the first. That's because the overall free energy change for the coupled reaction is equal to the sum of the free energy changes for each individual step. In this case, the delta G degrees for the coupled reaction will be negative 8 kcal per mole, making the overall pathway energetically favorable. Cells can therefore cause the energetically unfavorable transition X to Y to occur if an enzyme catalyzing the X to Y reaction is supplemented by a second enzyme that catalyzes the energetically favorable reaction Y to Z. In effect, the reaction Y to Z acts as a siphon, pulling the conversion of all of molecule X to molecule Y, and thence to molecule Z. Figure 321. For example, several of the reactions in the long pathway that converts sugars into CO2 and H2O are energetically unfavorable. The pathway nevertheless proceeds rapidly to completion, however, because the total delta G degrees for the series of sequential reactions has a large negative value. Forming a sequential pathway, however, is not the answer for all metabolic needs. Often, the desired reaction is simply X to Y, without further conversion of Y to some other product. Fortunately, there are other, more general ways of using enzymes to couple reactions together, involving the production of activated carrier molecules that can shuttle energy from one reaction site to another. We will see shortly how these systems work, but before we do, we shall look at how enzymes encounter their substrates and how enzyme performance is measured. Rapid diffusion allows enzymes to find their substrates. Enzymes and their substrates are both present in relatively small amounts in a cell, yet a typical enzyme can capture and process about 1,000 substrate molecules every second. This means that an enzyme must be able to release its product and bind a new substrate in a fraction of a millisecond. How can these molecules find each other so quickly inside the cell? Rapid binding is possible because motions are enormously fast at the molecular level. Because of heat energy, molecules are in constant motion and consequently will explore the space inside the cell very efficiently by wandering randomly through it, a process called diffusion. In this way, every molecule in a cell collides with a huge number of other molecules each second. As the molecules in a liquid collide and bounce off one another, an individual molecule moves first one way and then another, its path constituting a random walk. Figure 322. In such a walk, the average distance that each molecule travels as the crow flies from its starting point is proportional to the square root of the time it takes. That is, if it takes a molecule 1 second on average to travel 1 micrometer, it will take 4 seconds to travel 2 micrometers, 100 seconds to travel 10 micrometers, and so on. Thus, diffusion only works well for very short distances. To move molecules quickly over larger distances, cells need to rely on more active and directed methods of transport, processes that inevitably require an expenditure of cellular energy. The inside of a cell is very crowded. Figure 323. Nevertheless, experiments in which fluorescent dyes and other labeled molecules are injected into cells show that small organic molecules diffuse through the dense aqueous gel of the cytosol nearly as rapidly as they do through water. A small organic molecule, such as a substrate, takes only about 1/5 of a second on average to diffuse a distance of 10 micrometers. Diffusion is therefore an efficient way for small molecules to move limited distances in the cell. Enzymes and other macromolecules, however, diffuse through the cytoplasm much more slowly than do small molecules. In some cases, enzymes that interact with other proteins are actually held in close proximity to their partners by scaffold proteins that draw together sets of interacting proteins at specific locations in the cell. But even if they're not physically sequestered in one place, enzymes move much more slowly than small molecules, so we can think of them as standing still. Thus, the rate of encounter of each enzyme molecule with a small molecule substrate depends on the concentration of the substrate molecule. For example, some abundant substrates are present in the cell at a concentration of 0.5 millimolar. Because pure water is 55 molar, there is only about one such substrate molecule in the cell for every 10 to the power of 5 water molecules. Nevertheless, the active site on an enzyme molecule that binds this substrate will be bombarded by about 500,000 random collisions with the substrate molecule per second. For a substrate concentration tenfold lower, 0.05 millimolar, the number of collisions drops to 50,000 per second, and so on. A random encounter between the surface of an enzyme and the matching surface of its substrate molecule often leads immediately to the formation of an enzyme-substrate complex that is ready to react. A reaction in which a covalent bond is broken or formed can now occur extremely rapidly. Once one appreciates how quickly molecules move and react, the observed rates of enzymatic catalysis do not seem so amazing. When an enzyme and substrate have collided and snuggled together properly at the active site, they form multiple weak bonds with each other that persist until random thermal motion causes the molecules to dissociate again. These weak interactions can include hydrogen bonds, van der Waals attractions, and electrostatic attractions, as discussed in Chapter 2. In general, the stronger the binding of enzyme and substrate, the slower the rate of dissociation, as discussed earlier in this chapter. When two colliding molecules have poorly matching surfaces, few non-covalent bonds are formed and their total energy is negligible compared with that of thermal motion. In this case, the two molecules dissociate as rapidly as they come together. See Figure 232. This is what prevents incorrect and unwanted associations from forming between mismatched molecules, such as an enzyme and the wrong substrate. Vmax and K subscript M measure enzyme performance. To catalyze a reaction, an enzyme must first bind its substrate. The substrate then undergoes a reaction to form the product, which initially remains bound to the enzyme. Finally, the product is released and diffuses away, leaving the enzyme free to bind another substrate molecule and catalyze another reaction. See Figure 315. The rates of the different steps vary widely from one enzyme to another, and they can be measured by mixing purified enzymes and substrates together under carefully defined conditions. In such experiments, if the concentration of the substrate is increased progressively from a very low value, the concentration of the enzyme-substrate complex, and therefore the rate at which product is formed, initially increases in a linear fashion in direct proportion to substrate concentration. However, as more and more enzyme molecules become occupied by substrate, this rate increase tapers off until at a very high concentration of substrate, it reaches a maximum value, termed Vmax. At this point, the active site of all enzyme molecules in this sample are fully occupied by substrate, and the rate of product formation depends only on how rapidly the substrate molecule can be processed. For many enzymes, this turnover number is of the order of 1,000 substrate molecules per second, although turnover numbers between 1 and 10,000 have been measured. The concentration of substrate needed to make the enzyme work efficiently is often measured by a different parameter, the Michaelis Constant, K subscript M, named after one of the biochemists who worked out this relationship. An enzyme's Michaelis constant is a concentration of substrate at which the enzyme works at half its maximum speed, 0.5 Vmax, figure 324. In general, a low value of K subscript M indicates that a substrate binds very tightly to the enzyme, and a large value corresponds to weak binding. For a discussion of how we measure these parameters and how we can use them to model biochemical pathways and potentially to design better catalysts, see How We Know, pages 101 to 103. It is important to recognize that when an enzyme or any catalyst lowers the activation energy for the reaction Y to X, it also lowers the activation energy for the reverse reaction X to Y by exactly the same amount. See figure 312. The forward and backward reactions will therefore be accelerated by the same factor by an enzyme, and the equilibrium point for the reaction, and thus delta G degrees, remains unchanged. Figure 325. Activated carrier molecules and biosynthesis. The energy released by the oxidation of food molecules must be stored temporarily before it can be channeled into the construction of either other small organic molecules or the larger and more complex molecules needed by the cell. In most cases, the energy is stored as chemical energy in a small set of activated carrier molecules, which contain one or more energy-rich covalent bonds. These molecules diffuse rapidly throughout the cell and thereby carry their bond energy from the sites of energy generation to the sites where energy is used for biosynthesis and other needed cell activities. Figure 329. Activated carriers store energy in an easily exchangeable form, either as a readily transferable chemical group or as high-energy electrons, and they can serve a dual role as a source of both energy and chemical groups for biosynthetic reactions. The most important of the activated carrier molecules are ATP and two molecules that are closely related to each other, NADH and NADPH. Cells use activated carrier molecules like money to pay for reactions that otherwise could not take place. The formation of an activated carrier is coupled to an energetically favorable reaction. When a fuel molecule, such as glucose, is oxidized in a cell, enzyme-catalyzed reactions ensure that a large part of the free energy that is released by oxidation is captured in a chemically useful form, rather than being released wastefully as heat. Burning sugar in a cell allows you to power metabolic reactions, whereas burning a chocolate bar in the street will get you nowhere. Energetically speaking, it produces no metabolically useful energy. In living systems, this energy capture is achieved by means of a coupled reaction, in which an energetically favorable reaction is used to drive an energetically unfavorable one that produces an activated carrier molecule or some other useful molecule. Coupling mechanisms require enzymes, and they are fundamental to all of the energy transactions of the cell. The nature of a coupled reaction is illustrated by a mechanical analogy in figure 330, in which an energetically favorable chemical reaction is represented by rocks falling from a cliff. The energy of falling rocks would normally be entirely wasted in the form of heat generated by friction when the rocks hit the ground. Figure 330a. By careful design, however, part of this energy could be used to drive a paddle wheel that lifts a bucket of water. Figure 330b. Because the rocks can now reach the ground only after moving the paddle wheel, we say that the energetically favorable reaction of rocks falling has been directly coupled to the energetically unfavorable reaction of lifting the bucket of water. Because part of the energy is used to do work in B, the rocks hit the ground with less velocity than in A, and correspondingly, less energy is wasted as heat. The energy saved can be used to do useful work. Figure 330c. Analogous processes occur in cells, where enzymes play the role of the paddle wheel in Figure 330b. By mechanisms that will be discussed in Chapter 13, enzymes couple an energetically favorable reaction, such as the oxidation of foodstuffs, to an energetically unfavorable reaction, such as the generation of an activated carrier molecule. As a result, the amount of heat released by the oxidation reaction is reduced by exactly the amount of energy that is stored in the energy-rich covalent bonds of the activated carrier molecule. The activated carrier molecule, in turn, picks up a packet of energy that is large enough to power a chemical reaction elsewhere in the cell. ATP is the most widely used activated carrier molecule. The most important and versatile of the activated carriers in cells is ATP, adenosine 5' triphosphate. Just as the energy stored in the raised bucket of water in Figure 330b can be used to drive a wide variety of hydraulic machines, ATP serves as a convenient and versatile store or currency of energy to drive a variety of chemical reactions in cells. As shown in Figure 331, ATP is synthesized in an energetically unfavorable phosphorylation reaction in which a phosphate group is added to ADP, adenosine 5' diphosphate. When required, ATP gives up this energy packet in an energetically favorable hydrolysis to ADP and inorganic phosphate, P subscript I. The regenerated ADP is then available to be used for another round of phosphorylation reaction that forms ATP, creating an ATP cycle in the cell. The energetically favorable reaction of ATP hydrolysis is coupled to many otherwise unfavorable reactions through which other molecules are synthesized. We shall encounter several of these reactions later in this chapter and see exactly how this is done. Such hydrolysis reactions often involve the transfer of the terminal phosphate in ATP to another molecule, as illustrated in Figure 332. Any reaction that involves the transfer of a phosphate group to a molecule is termed a phosphorylation reaction. Phosphorylation reactions are examples of condensation reactions. See Figure 225 and are involved in many important cellular functions. They activate substrates, they facilitate the exchange of chemical energy, and they help to control cell signaling processes. ATP is the most abundant energy carrier in cells. It is used, for example, to supply energy for many of the pumps that transport substances into and out of the cell, discussed in Chapter 12. It also powers the molecular motors that enable muscle cells to contract and nerve cells to transport materials from one end of their long axons to another, discussed in Chapter 17. Why evolution selected this particular nucleotide over the others as the major carrier of energy, however, remains a mystery. Energy stored in ATP is often harnessed to join two molecules together. We have previously discussed one way in which an energetically favorable reaction, Y to Z, can be coupled to an energetically unfavorable reaction, X to Y, so as to enable it to occur. In that scheme, a second enzyme catalyzes the energetically favorable reaction, Y to Z, pulling all of the X to Y in the process. See Figure 3-1. This mechanism is not useful, however, when the required product is Y and not Z. A common type of reaction that is needed for biosynthesis is one in which two molecules, A and B, are joined together to produce AB. In the energetically unfavorable condensation reaction, AH plus BOH to AB plus H2O. ATP hydrolysis can be coupled indirectly to this reaction to make it go forward. That is, energy from ATP hydrolysis is first used to convert BOH to a higher energy intermediate compound, which then reacts directly with AH to give AB. The simplest possible mechanism involves the transfer of a phosphate from ATP to BOH to make BOPO3, in which case the reaction pathway contains only two steps. 1. BOH plus ATP to BOPO3 plus ADP. 2. AH plus BOPO3 to AB plus P subscript I. Net result, BOH plus ATP plus AH to AB plus ADP plus P subscript I. The condensation reaction, which by itself is energetically unfavorable, has been forced to occur by being coupled to ATP hydrolysis in an enzyme-catalyzed reaction pathway. Figure 333a. A biosynthetic reaction of exactly this type is employed to synthesize the amino acid glutamine, as illustrated in Figure 333b. We will see later in the chapter that very similar but more complex mechanisms are also used to produce nearly all of the large molecules of the cell. NADH and NADPH are important electron carriers. Other important activated carrier molecules participate in oxidation and reduction reactions and are commonly part of coupled reactions in cells. These activated carriers are specialized to carry both high-energy electrons and hydrogen atoms. The most important of these electron carriers are NAD+, nicotinamide adenine dinucleotide, and a closely related molecule, NADP+, nicotinamide adenine dinucleotide phosphate. NAD+ and NADP+ each pick up a packet of energy in the form of two high-energy electrons plus a proton, H+, becoming NADH, reduced nicotinamide adenine dinucleotide, and NADPH, reduced nicotinamide adenine dinucleotide phosphate, respectively. These molecules can therefore also be regarded as carriers of hydride ions, the H+, plus 2 electrons, or H-. Like ATP, NADPH is an activated carrier that participates in many important biosynthetic reactions that would otherwise be energetically unfavorable. NADPH is produced according to the general scheme shown in Figure 334. During a special set of energy-yielding catabolic reactions, a hydrogen atom and two electrons are removed from the substrate molecule and added to the nicotinamide ring of NADP+ to form NADPH. This is a typical oxidation-reduction reaction. The substrate is oxidized and NADP+ is reduced. The hydride ion carried by NADPH is given up readily in a subsequent oxidation-reduction reaction, because the ring can achieve a more stable arrangement of electrons without it. In this subsequent reaction, which regenerates NADP+, the NADPH becomes oxidized and the substrate becomes reduced, thus completing the NADPH cycle. NADPH is efficient at donating its hydride ion to other molecules for the same reason that ATP readily transfers a phosphate. In both cases, the transfer is accompanied by a large negative free energy change. One example of the use of NADPH in biosynthesis is shown in Figure 335. The difference of a single phosphate group has no effect on the electron transfer properties of NADPH compared with NADH, but it is crucial for their distinctive roles. The extra phosphate group on NADPH is located far from the region involved in electron transfer. See Figure 334b. However, it serves to give a molecule of NADPH a slightly different shape from that of NADH, making it possible for NADPH and NADH to bind as substrates to different sets of enzymes. These two types of carriers are thereby used to deliver electrons or hydride ions to different destinations. Why should there be this division of labor? The answer lies in the need to regulate two sets of electron transfer reactions independently. NADPH operates chiefly with enzymes that catalyze anabolic reactions, supplying the high-energy electrons needed to synthesize energy-rich biological molecules. NADH, by contrast, has a special role as an intermediate in the catabolic system of reactions that generate ATP through the oxidation of food molecules, as we discussed in Chapter 13. The genesis of NADH from NAD+ and that of NADPH from NADP+ occur by different pathways and are independently regulated so that the cell can adjust the supply of electrons for these two contrasting purposes. Inside the cell, the ratio of NAD+ to NADH is kept high, whereas the ratio of NADP+ to NADPH is kept low. This arrangement provides plenty of NAD+ to act as an oxidizing agent and plenty of NADPH to act as a reducing agent, as required for the special roles in catabolism and anabolism respectively. Cells make use of many other activated carrier molecules. Other activated carriers also pick up and carry a chemical group in an easily transferred high-energy linkage. Table 3.2. For example, FADH2, like NADH and NADPH, also carries hydrogen and high-energy electrons. See Figure 13.12. Coenzyme A, on the other hand, can carry an acetyl group in a readily transferable linkage. This activated molecule, called acetyl-CoA, acetyl coenzyme A, is shown in Figure 3.36. It is used, for example, to add 2 carbon units in the biosynthesis of the hydrocarbon tails of fatty acids. In acetyl-CoA and the other carrier molecules in Table 3.2, the transferable group makes up only a small part of the molecule. The rest consists of a large organic portion that serves as a convenient handle, facilitating the recognition of the carrier molecule by specific enzymes. As with acetyl-CoA, this handled portion very often contains a nucleotide. This curious fact may be a relic from an early stage of cell evolution. It is thought that the main catalysts for early life forms on Earth were RNA molecules or their close relatives, and that proteins were a later evolutionary addition, discussed in Chapter 7. It is therefore tempting to speculate that many of the carrier molecules that we find today originated in an earlier RNA world, where their nucleotide portions would have been useful for binding these carriers to RNA enzymes. In addition to the transfer reactions catalyzed by the activated carrier molecules, ATP, transfer of phosphate, and NADPH, transfer of electrons and hydrogen, other important reactions involve the transfers of methyl, carboxyl, and glucose groups from activated carrier molecules for the purpose of biosynthesis. The activated carriers are usually generated in reactions coupled to ATP hydrolysis. Figure 337. Therefore, the energy that enables their groups to be used for biosynthesis ultimately comes from the catabolic reactions that generate ATP. Similar processes occur in the synthesis of the very large molecules of the cell, the nucleic acids, proteins, and polysaccharides, which we discuss next.